All right, so the different sections that we're going to describe are passive transport and active transport. And in passive transport, it is diffusion, facilitated diffusion, osmosis. And then for active transport, it is protein pumps, endocytosis, and exocytosis. All right, so we're going to first start with passive transport and diffusion. And what we have here is like all molecules, atoms, and it's in like
The key ring is the cell wall, and the paper is the molecules.
All right, and what happens is they're in a high area of concentration, and then they like kind of like spread out to form into lower areas of concentration to form an equilibrium. All right, so next we're going to describe facilitated diffusion, and we're going to use the same representative here. And then what we're going to use is this is going to be our protein channel. And what happens in facilitated diffusion is the molecules, and this is a piece of papers, they're going to like cross and everything.
And then go into the protein channel.
You know what I'm saying? Alright, next we're going to describe osmosis, and we're going to use the same diagram again, and it's going to be an area of high concentration
Of water right here.
So paper is going to represent the waters, and then what they're going to have to do is they're going to have to move so they have an equilibrium, so it's 3 and 3. So the higher concentration would have to move to the lower concentration, so there's an equilibrium.
Next, we're moving on to active transport, and first we're going to talk about protein pumps.
Alright, and then for the diagram, these are going to be the channels that the protein pump's going to go through. And then this is obviously the cell and the cell membrane. And what happens is the protein pump, this piece of paper, will go through, and as it's going through, two other, like, they could be bacterias, or it could be other, like, potassiums go through, and they'll pump potassium into the cell. And then when it leaves, it'll do the exact same thing, and then it'll go back through and, like, exit it, so it's going in and out of it.
Now we're moving on to endocytosis. The two major parts of it are phagocytosis and pinocytosis.
Alright, and so I'm going to first describe pinocytosis, and what pinocytosis is as pinocytosis, this is the pinocytosis, moves in with a vesicle, and then when it does is, as it's moving in, it joins up with a lysosome, and then what it does is it hydrolyzes and it breaks down other foods and particles, and then phagocytosis is when, here this could be the phagocytosis, so when the phagocytosis moves in and it engulfs smaller particles, and what those smaller particles do, they could be like proteins or nutrients, or they could be bacteria, and what they do is it'll engulf them, and then it can either move them throughout the body as nutrients and things for other body parts, or it can expose of them if they're bacteria.
Now, lastly, we're moving on to exocytosis.
And exocytosis, this is going to be the vesicle that we're going to talk about, and this is going to be the waste particles. What happens is the vesicle takes the waste particles, and what it does is it fuses them with the plasma membrane, and then the plasma membrane then, as it's like in the plasma membrane, it then goes outside of the cell, so it's like outside of the cell then, and it disposes of it.